So this video is covering protein absorption and digestion. So our protein enters our oral cavity over here. So it doesn't really, nothing really happens much inside the oral cavity. So it's chilling, enters, goes through the esophagus. I haven't got any annotations. Now it enters into our stomach. Our stomach is the most important part, or probably like the first important part in terms of protein digestion. So first, let's look over here. We've got different cells lining the stomach, like this in the stomach lining. We've got our chief cells and we've got our parietal cells. Our chief cells, they release pepsinogen. Pepsinogen, OGEN, is inactive. And then our parietal cells, they release HCl. HCl has two roles. One of HCl's roles is to denature our protein. So here we've got protein over here. That's a more scruffier diagram, but that's our protein. HCl works to denature that via the mechanism I'll briefly explain afterwards. And HCl also activates pepsinogen into pepsin. So that's our little cells on the side. Now let's look onto our protein. So our protein is entered into our stomach. Now HCl, HCl is an acid, so of course that's one of the things which makes the stomach so acidic. It produces the H+, so our H+ is there just chilling over here. And what happens is H+ is positive. However, our proteins are slightly negatively charged. So, and you can also see here we have the NH2 group, we have the COH group at the top. So yeah, like I said, the proteins are slightly negatively charged. So what the H+ does is it tries to steal the electrons from the proteins, and this causes it to like unravel, and the whole process is just called denaturation. And then that causes a linear like polypeptide kind of thing to form. Now our linear thing comes over here. Next thing we're going to look at is going to be our pepsinogen. So firstly, we've broken it down, we've made it simpler. I like the analogy that Dr. Matt and Dr. Mike YouTube channel used, was they said a ball of yarn, it's hard to like cut if you just try and cut a ball of yarn. However, if you just make it like straight into pieces of rope or whatever, or I don't know, it'll be easier to cut. I like the analogy, that works for me. So next thing we're looking at is HCl's second function, which I said first function was to denature the protein. Second function was to activate pepsinogen. Sorry, to activate pepsin. No, yeah, to activate pepsinogen. So here we have our pepsinogen, and that is inactive. So HCl activates it. Now pepsin is our molecular scissors. Comes over here, snips that apart, makes that into smaller chains. So here we've got our bit scruffy polypeptide chains. A bit easier here, and over here like the dots all represent like amino acids because this is a whole chain. So over here now, important thing to remember is if we link back, if you watch the previous video on what's it called, I think it was carbs, we're talking about the cells that line our duodenum. So that is our enteroendocrine cells, the type of cells that line it. And what happens is when our proteins, or like the more linear version, enters into our duodenum, I'll move back a tiny bit. It causes the release of CCK. CCK, once again, I'll probably mix it up. I think it's cholecystokinin. That goes over there. Sorry, yeah, enteroendocrine cells release CCK. CCK goes over here into the pancreas, next to the pancreas. CCK triggers the release of pancreatic juices. So let's follow the red line over here. And the pancreatic juices contains enzymes. For carbs, it was different ones. But for proteins, these are different enzymes it releases. Important thing to bear in mind that these enzymes are currently inactive. We've got trypsin ogen, chymotrypsin ogen, and pro-elastase and pro-carboxypeptidase. The pro here signify that they are inactive. So now another important thing to note is trypsinogen, chymotrypsinogen, and pro-elastase. These are all endopeptidases, meaning they cut through the middles. However, exopeptidase is more like it cuts the terminal bonds, so it would form like a longer chain as well as a single amino acid. Anyway, these are all inactive. Next thing we'll be looking at is how can we activate these to form like the enzymes. The way we do that is we go over into the lining. I kind of was running out of space, so I just crammed it in there. We've got our enterokinase cell. Our enterokinase cells would firstly activate the trypsinogen to form trypsin. Now what trypsin does is trypsin then activates our chymotrypsinogen, proelastase, and procarboxypeptidase until they, I don't know, form like chymotrypsin, elastase, and carboxypeptidase. Like I mentioned before, carboxypeptidase is a exopeptidase, and there's two types. We've got our A and our B. What significance I have, I don't know. I'm just remember that off Dr. Johnson's slides. Anyway, these trypsin activates all of those. Now they start getting to work on our protein, our polypeptide chain. Now they're getting to work on our polypeptide chain. My brain's gone blank. Now I remember. Important thing to bear in mind here is what they are. So here, procarboxypeptidase is an exopeptidase. Exo meaning terminal. Now what I mentioned earlier was that means it forms the longer chain and it forms your singular amino acids. So, sorry, ignore, I think I said procarboxypeptidase. I'm talking about the active version of this. So we're talking about carboxypeptidase. And what that does is that produces amino acids. Now, however, we've got our endopeptidases, which will produce our di and tripeptides. Reason being is because they're cutting through the middle. So there's still, like, for example, if I cut through here, you got 2 on this side, 2 on that side, for example. So yeah, now we're over here. So we're left with our amino acids, and over here we've got our dipeptides and our tripeptides. But we want to try and break this down as much as we can. For this reason, now we go to, I don't know, this part just sticks with me for some reason, our brush border cells. This is our brush border cells. Let me zoom back a tiny bit so you can see that context. Ignore all the green. Our brush border cells, that was my bad, lol. Anyway, brush border cells of the small intestines, they release our brush border enzymes. Let's go down the arrow. So remember, we have our amino acids, which they're already broken down, but we want to break our di and tripeptides, our di and tripeptides into smaller ones. So the brush border releases the brush border enzymes. In this context, we've got our dipeptidase and our aminopeptidase. So our aminopeptidase, it kind of just chops the ends. So let's look at this in the context of a tripeptide over here. If we cut the end off, we've got one amino acid here, and we've got a dipeptide here, which is good because then our dipeptidase gets to work, and that cuts the dipeptides into amino acids. So, for example, if you look here, it's literally a dipeptide. Cut it through the middle, we've got 2 amino acids. Over here, let's say, for example, our aminopeptidase, chop the end off, we've now got 2 left. Cut that in the middle with our dipeptidase, we've now got singular amino acids. So now we've got the singular amino acids. Let's now look into how we're going to get those into our bloodstream. So we've got enterocytes. Enterocytes is talking about these like cell kind of things. Now, same concept as before. First things first, let's go all the way over here. We've got our sodium potassium ATPase pump. So same concept as before. 3 sodiums out, 2 potassiums in. 3 sodiums out, what it does is it reduces the concentration of sodium inside our enterocytes. Beneficial, reason being is because then we've got our sodium, let's say we've got it ingested in our food, comes over here, sodium's in our diet. Sodium wants to enter the cell because high concentration to low concentration. Now, this enters the cell via SGLT channel. Sorry, I think I blocked the thing. SGLT. Now, the interesting thing here is when sodium enters, and we can also allow for amino acids also to enter. This is via secondary active transport, also known as co-transport. I think that's just what I call it. And yeah, so sodium passes through, and our amino acid also passes through. That is how just the amino acids pass through. However, at times we also there may be issues with this, and they may not fully break it down into amino acids. In such case, then we've got our dipeptides and our tripeptides. Now, question is, how are we going to get those into our enterocytes? The way we're going to do that is through this. This is called a pepT1. If what I'm saying is wrong, I'm going to correct it in the comments afterwards. I've searched that, inshallah that's correct. I haven't seen it on his slides though, so I don't know. Anyway, same concept, I'm inshallah pretty sure is correct. H plus is trying to enter through here, through the channel. And whilst that is happening, then we've got our amino acids. They can also pass through with it, as well as our di and tripeptides. Important thing to mention, this is coupling with H plus now, not with sodium. Not with sodium. So with H plus, we've then got our di and our tripeptides. They're also entering our enterocyte. Now, what happens if there's some big issues with this or whatever, and we didn't even break it down to the di and the tripeptides, and we broke it down into a polypeptide chain. Let's say we've got a polypeptide chain over here. Then what can happen is it can enter the enterocyte via endocytosis or phagocytosis specifically. We've got the invagination, goes like that, seals it, puts it into the enterocyte. Now in our enterocyte, we've now got our amino acids, which are perfect, go straight through. But we've also got our dipeptides, which I kind of crossed out the I by accident. And we've got a tri, which looks like an M because, I don't know, I just can't write small. And we've also got our polypeptide chains. So we want to break these down into amino acids. The way this happens is our enterocytes also contain peptidases. And these peptidases, like I mentioned, are molecular scissors. They go snip, 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 cut those up into our amino acids. Next, we're left with our amino acids. They then enter into the bloodstream via facilitated diffusion. I've searched what the name of this is, and I don't really know. If I find out, then I'll put it in the comments. And yeah.